Here we go. We're there finally. Okay, just starting off, just a little quick plug for my my website. As you can see, www.biologycarol.co.uk. I have got the full range of A-level teaching videos for both OCR and AQA on there. And they'll cost you to subscribe £100 for a whole year. Now, I'm hoping that that is sounding really reasonable, and that it is, and that's because that is probably less than a couple or three maximum tutoring sessions. So hopefully you'll agree it's reasonable value for money. If you just need the AS ones, it's half the price. And because my aim is to make A-level biology accessible to all students, and even those who can't afford tutoring, I give you 10% commission on any of your friends' purchases. So, and there's a freebie, so go and have a look at it anyway, see what you think. So here we are, we're doing the AQA AS Paper 2 from 2017. And let's just crack on because I have a deadline for 6 o'clock to get this done by. So question 1, glycogen and cellulose, both carbohydrates. Describe two differences between the structure of a cellulose molecule and the glycogen molecule. We're talking differences. You've got to pick a feature and say what it's like for one and what it's like for the other. So here are the two things that I wrote. First of all, I talked about the glucose molecules that make up each of the polysaccharides. Cellulose beta glucose, glycogen alpha glucose. And then I talked about whether the molecule is straight chained, linear, or branched. So there were my two answers. And there's the mark point, and here are the examiner's comments, and here is where I got the 2 marks. Notice that linear means the same thing as straight chained. Worth noting, if you're watching this in non-live stream land, do just stop the video and have a go at the questions yourself before I show you what I would have written. And just to say that I number my ticks according to the mark points in the mark scheme, and I put the tick at exactly the point in my answer where I think I earned the mark. So hopefully that will be helpful. Starch is a carbohydrate, often stored as in plant cells. Describe and explain 2 features of starch which make it a good storage molecule. These answers need to have a particular feature, and then the next word needs to be because, so, as a result of, so that, those kinds of things. Because after those words, you're then explaining. So, I've said it's insoluble, that's the feature or the property. And so it doesn't affect the cell's water potential. And it is coiled because I'm thinking of the amylose molecule, which is the one emphasized in the AQA syllabus. So it is compact. Here's the mark scheme. Here are the examiner's comments. Notice that the psi symbol is allowed for water potential. And therefore I got mark point 1 and mark point 2. Notice that they allowed references to amylopectin if that's what you'd been taught about and that's what you put in your answer. You could talk about the branched molecules, but those are the ones most obvious to me. Tick the box that identifies the test that you would use to show the presence of starch. And that is, of course, the iodine test, because it goes from that kind of mid-blue colour to a blue-black colour for a positive test of starch. Hopefully that is relatively straightforward. Now we get a section through a plant tissue at a magnification of ×500. Calculate the actual diameter of the starch grain between points A and B. Now, in order to be able to do this, I actually printed off the page at 100% because this was as close as I would get to what the actual page in the exam booklet would look like. So I then measured carefully using my ruler in millimeters. I measured the distance and it ended up being 20 millimeters. So 20 millimeters, and here's my tip to you, always work in millimeters. Don't be tempted to put 2 centimeters and then try and do the conversion into micrometers. Always work straight into millimeters, because then the conversion is always times by 1,000. I think it's just simpler. So we have 20 millimeters is the same as 20,000 micrometers. But of course, that's the size of the picture, the image. So we need to divide it by the number of times that image has been magnified to get the actual distance across that starch grain. And that is 40 micrometers. Sorry, I left this examiner's comment up and I got the right answer. What type of microscope has been used? And give one piece of evidence from the picture to explain, to support your answer. So I'm looking at that and going, everything is in 3D and we are seeing the surfaces of structures, therefore it's got to be a scanning electron microscope. And I happened to put that it showed a 3D picture, and that was the answer that they wanted. They wouldn't accept, notice, that it was showing only the surfaces of structures, because of course, actually you were cutting through cells here. So that wasn't actually a valid answer. Hope you understand that. So even though we're only seeing the surfaces of things, you have to be incredibly careful in your wording. 3D image, by far the safest answer. Hi everybody, lovely to see you. So just welcome everybody, and those of you in live stream land and those of you who are just watching this as a video. I tend to go through this quite quickly because that then makes the video shorter. I don't want to kind of waste your time, and obviously play me on double speed if you possibly can. Question 2, cells of the beetroot contain a red pigment, but a student investigated the effect of temperature on the loss of red pigment from the beetroot cells. He put discs, you'll be familiar with this experiment, put discs into tubes containing water. He maintained each tube at a different temperature, and after 25 minutes, he measured the percentage of light passing through the water in each tube. He put the same volume of water in each tube. Explain why it was important that he controlled this experimental variable. Well, this is what I put. Because the red pigment would be diluted to the same extent for each temperature. Maybe I should have said that, for each temperature. And then you're kind of going, well, why is that important? Well, because the concentration of the red pigment, notice I've used the term directly proportional to the percentage of light passing through it. And in fact, that is not correct. Here am I suddenly realizing, not to the percentage of light passing through it, that actually isn't correct. So actually, I'm now realizing, sorry, here's the mark scheme, and I gave myself 1 mark. So why isn't this correct? Because if the concentration of red pigment was 0, the percentage light passing through it wouldn't also be 0, it would be 100%. I was thinking of absorbance. If the percentage of red pigment was 0, the absorbance would be 0. It would have been completely accurate for me to say that it was directly proportional. This is not directly proportional. So therefore, this sentence is scientifically wrong. And therefore, I gave myself 1 mark here, but I'm taking it away. Notice what you had to write. I would never have written mark point 1. I would have written what I wrote and hopefully realized that I've actually got this wording wrong. And I missed this out. I thought I'd written enough for 2 marks. Now, here's the thing. Notice that when I'm writing on the slides, I am writing with relatively huge writing in comparison to the number of words that you would get on a line when you wrote in an exam paper. So I kind of run out of space far quicker than you would. I wasn't getting the prompt, oh my goodness, I've got lots of space left. Not that that should be a major thing for you to be trying to do for every exam paper. Please, so here am I balancing a little bit of an excuse for me, but actually don't use them, don't feel obliged to fill every number of lines. What is far more important is that when you are answering the question, you are either saying, for a 2-mark question, have I given 2 decent points? Or even better, can I add a third in the time that I have got to make it more sure that I get the 2 marks? I kind of thought I might have written enough for 2 marks, but as you can clearly see, I'd only written enough for 1 if I'd got it right, and I missed out the 2nd mark. So describe a method the student could have used to monitor the temperature of water in each tube. Well, that of course is using a thermometer, but to monitor the temperature, you've got to take repeated readings of the temperature. You can't just put a thermometer in the tube. You're not monitoring anything unless you are constantly, in effect, looking at the thermometer. In other words, taking readings. There you go. So hence I've written that and I got the mark because the repeated readings was important. Here's the student's results. Draw a suitable curve. Now notice you can either draw straight ruled lines dot to dot, or you could draw a free form line of best fit. Notice for a line of best fit, your line actually shouldn't pass through every point. The line should pass through showing the trend of the data. And as a general rule, you should have equal numbers of points on either side of the line. And if you really want to make your line of best fit good, the distance from the line of the points on one side should be approximately equal of the distance of the points on the other side. There's how you draw a line of best fit. Nearly impossible for me to do on this screen without it looking almost ridiculous. I decide to do ruled lines from point to point. Notice they are not extrapolated beyond the data points from the experiment. Really crucial. So I got the mark, no extrapolations, notice, and shouldn't look sketchy. The decrease in the percentage of light passing through the water between 25 and 60 degrees is caused by the release of red pigment from the cells of the beetroot. Hopefully that makes sense. The redder the solution is, the less light that is going to pass through it. Suggest how the increase of the temperature caused the release of the red pigment. So here am I kind of going, flipping heck, this is a 2-mark question. And what I'm actually wanting to do is to explain the whole shape of the curve, that actually the percentage of light passing through dropped as more pigment was released, because of course, as the temperature went up to about 40 degrees, the phospholipids in the membrane were increased in their kinetic energy, and therefore there was some leakage of the pigment. Between 40 and crumbs, 44 degrees, something like that, this is where it is likely that the membrane ruptured. It went absolutely, was damaged, and therefore you got a great increase in the amount of red pigment escaping. So, and it plateaus off as the temperature increases more. So I tried to encapsulate this with my very large writing and relatively few lines. So this is what I wrote. And here is the mark scheme. Now, I've given myself mark point 1 here because membrane rupturing, I believe, is the same as saying that there is damage to the membrane. And I kind of hope that I might get mark point 3 because I'm talking about this increased kinetic energy, i.e. increased fluidity of the phospholipid bilayer. I'm not sure I would have got mark point 3. So, you know, we could take that out of the equation if we wanted to. Question 3. Most human cells contain 2 copies of each gene. However, there may be up to 15 copies of the gene for amylase. That is the enzyme that breaks down starch into maltose. Scientists investigated the number of copies of the AMY1 gene in individual peoples in 2 populations, one high starch diet population and one low starch diet population. And here are the results. Describe what their results show. So the results to me, so the first thing that I do is that I look to see whether the range is different, and then I notice that actually the modal number for the high starch population, modal meaning the value, i.e. the number of copies of the gene at which the frequency, which has the highest frequency. The modal number for the high starch population is higher than the modal number for the low starch population. So here is what I wrote. Here, the high starch population, the range was greater than the low starch population. The modal number for AMY1 genes was higher, 6, in the and also higher compared to the low starch population when it was 4. But I also noticed that actually the percentage of individuals, as it were, the frequency for the modal number of the low starch population was a higher percentage than that of the high starch population. Here's the mark scheme, examiner's comments, and this is where I got the marks. I got mark 0.4. I got mark 0.5. Now, I have to say, reading the mark scheme, I actually agree more. I should have said mark 0.2, that actually the ranges were really similar, rather than saying one was larger than the other. I actually think mark 0.2 is the more valid comment, having seen the mark scheme. And those were the only 2 marks that I got. Notice that I didn't get, there was nothing for this saying this. So I didn't get all of the marks on that one. Multiple copies of the AMY1 gene is an adaptation to a high starch diet. Use your knowledge of protein synthesis, transcription and translation, and enzyme action to explain the advantage of this adaptation. So hopefully this answer makes sense. Each copy of a gene codes for a protein. If there are more copies of the gene, then there will be greater transcription. Yeah, if you only got 1 copy, there's 1 lot of transcription going on. If you've got 2 lots, transcription can be happening at the same time, multiply. So there will be greater transcription and translation of the gene into the enzyme. And of course, that means there will be, so here am I now applying my enzyme action knowledge, because I know that enzyme concentration increased enzyme concentration increases the rate of the reaction. But here the reaction is digestion, starch to maltose. So this is therefore what I wrote. Here's the mark scheme. Here are the examiner's comments, and here is where I got the marks. Okay, question 4. Do feel free to jot down any questions in the chat as we go along. There is a little bit of a delay, but I will keep looking at my second screen to see if you've got any questions. Really happy to answer. Question 4, scientists investigated how the concentration of protein in blood plasma changes in people between the ages of 65 and 95. Figure 4 shows the results, and the bars are plus or minus 1 standard deviation. What is the difference between males and females in the fall of mean concentration of protein in blood plasma between 60 and 95 years? Well, I don't know about you, I had to read that question twice to get my head around what the maths was that they wanted me to do. What is the difference in the decrease between males and females, going from 60 to 95 years. So we've got to work out the male decrease first, and that is 5 grams per decimeter cubed. Female decrease goes from 71 down to 60, and that's 11. The difference, maths ain't hard, 6. Here was all about reading the question carefully and working out what they wanted you to do, because clearly this maths isn't tricky. Use figure 4 to calculate the rate of change of the mean concentration of protein in the blood plasma of males between the ages of 60 and 95. Well, rate, I always say, if you don't like the word rate, substitute it for the word speed, because speed is a very particular type of rate, because rate is about the change that happens divided by the time that it's taken. Speed is the change in distance. How far have you gone divided by the time that it's taken? And of course, if you think of speed, hopefully you will think of the units miles per hour. And hopefully that reminds you that it's dividing the change, the number of miles in speed in case, divided by the time that it took to do those miles. Anyway, long-winded way of reminding you about change and how to do the calculation. Now, we've already done the change because, of course, it's the decrease, which is 5, and it happens over 35 years. And therefore, that is your calculation, 5 divided by 35, which equals 0.14. Now, I put the minus in front because I wanted to indicate that I knew that the change was a decrease, and therefore it was a negative change, a minus change. Here's the mark scheme and the examiner's comments. Ignore the signs, didn't need the sign, but I got the 2 marks. What can you conclude from figure 2 about the effect of aging on the mean concentration of protein in the blood plasma in males and females? Here is what I wrote. Female blood protein starts higher than males at 60 degrees, but has a greater decrease over the next 35. The difference is only significant at 95 years. This is the only point at which the standard deviations overlapped. That they didn't overlap. Oh, look at that. I missed out the word didn't. Okay, all right, okay, right, just realised that. Here's the mark scheme. Here's the no examiner's comments. I've given myself mark 0.2. Now, clearly, I can't give myself mark 0.2 because I actually wrote the wrong thing by mistake, but that's not the examiner's fault. The mark point that I'm really kicking myself for not saying clearly or more clearly was that there was a decrease in both male and females. Now, I think I have implied it in my first sentence. I'm not absolutely sure whether I'd have got the mark. I've chosen in this situation not to give it to myself. The scientists measured the absorption of each sample of blood plasma using a colorimeter. They used the calibration curve, to find the concentration of protein in samples of blood plasma. Describe how the scientists could obtain data to describe how they would obtain the data to produce a calibration curve and how they would use the calibration curve on an unknown sample of protein in plasma. So, first of all, as my answer says, they would have had to have had a whole range of known protein concentrations in blood plasma, and they would have to measure the absorbance of all of those samples. They would have to then plot that on a graph. On the x-axis would be the protein concentration, and on the y-axis would be the absorbance. And then with that unknown sample, you'd measure the absorbance. Oh yeah, and you'd have to draw a line of best fit, whatever. Then with the unknown sample, you would measure the absorbance and you would read off from the graph to find out what the protein concentration was. So there's the basic method. Can I just say, as I was writing this answer, I didn't have the mark. I wasn't reading the mark scheme. As I was writing this answer, I was literally getting to plotted on a graph. And I immediately realized, oh, crumbs, I could say which way the axis went round. And I just decided to put it into brackets as a quick piece of extra information. And lo and behold, that was what got me one of the marks, that's extra bit of information. And there's the difficulty with A-level. It is tricky to know the level of detail sometimes that is required to get the mark. Because this question was talking about how you would do the method, I thought that it made sense for me to include that level of detail about the graph. But it was touch and go as to whether I thought I was going to add it. And I'm pleased I did, clearly. Here's the examiner's comments, and here is where I got the marks. Marks 1 and 2 there, mark 3 there, because you had to even talk about drawing a curve, you had to work quite hard for mark point 3, and then mark point 4. Now, I got all 4 marks. Clearly, I'd have only got 3 because that's the tariff of the question. Older people are more likely to suffer from infectious diseases. Suggest how this might be linked to a decrease in blood protein as they get older. Well, now we're thinking about what kind of proteins in the blood help with overcoming infectious diseases. And hopefully you thought antibodies like I did. Because of course, those decreasing protein levels could be because of decreasing antibody levels. So I got that mark. Figure 5, oh, here we are on question 5. Figure 5 shows the oxyhemoglobin dissociation curves for both fetal hemoglobin and adult hemoglobin. Explain how changes in the shape of hemoglobin, you had to read this question carefully to have any hope of getting the marks. Explain how changes in the shape of hemoglobin result in the sigmoid-shaped curve. And I desperately wanted to explain every part of the curve, but I noticed that the tariff was 2 marks. I didn't know where the marks were. Because, where are we? Adult hemoglobin. This part of the graph is because it's actually quite tricky for the first oxygen molecule to bind to the hemoglobin. However, that first oxygen molecule causes a change in shape of the tertiary structure of the hemoglobin. And therefore, that makes oxygen molecules 2 and 3 far more easy to bind, which is why you get this sharp gradient here. And then finally, the 4th oxygen is harder to bind again. Here's the mark scheme. Now, I got the marks, but I thought that was hard to know what to write for the 2 marks. At birth, 98% of hemoglobin is fetal hemoglobin. And by the age of 6 months, all of that has been exchanged for adult hemoglobin. Use figure 5 to explain why this change is an advantage for the baby. I went down the wrong route in my answer. So this is what I wrote, which I thought was perfectly reasonable. Fetal hemoglobin has a higher affinity for oxygen than adult hemoglobin. Before birth, this is the only way that the baby is going to get any oxygen, because the fetal hemoglobin, because it has a greater affinity, it takes the oxygen from the maternal adult hemoglobin at the placenta. After birth, breathing means that in effect the fetal hemoglobin isn't required, only adult hemoglobin is needed. Here's the mark scheme. Didn't get mark point 1. I prefer this variation on mark point 1. Because I'm kind of going, well, fetal hemoglobin is lower practically every point except for 0 and, you know, close to 100. Has a lower affinity affinity for oxygen at partial pressures found in tissues. And you kind of go, well, that's fair enough at the partial pressures, you know, at the placenta. But I wouldn't have written it. I know I wouldn't have. Even though if the question, some other questions that I've seen for oxygen dissociation curves, it is clear from the question that you want to be referring to the partial pressure. And clearly this was the case here too. So there's a little tip. Oxygen dissociation curves, if you know you're going to be talking about the different affinity of hemoglobins, I would now be putting in the partial pressure at which that applied. I didn't even go anywhere near getting mark point 2. So I didn't get any marks for this answer. Sickle cell disease is caused by the production of faulty adult hemoglobin. This results in less oxygen being transported. And scientists investigated the use of hydroxyurea to treat babies with sickle cell disease because hydroxyurea changes the percentage of fetal hemoglobin in the blood. And they did this experiment on 122 babies. They treated them with hydroxyurea for 41 months, and they tested the mean concentration of fetal hemoglobins in those babies before and after the treatment. And the scientists concluded that the treatment with hydroxyurea would increase the concentration of oxygen in the blood of babies with sickle cell disease. Suggest why Figure 5 and Table 1 support this conclusion. So I started with Table 1. So notice that in effect I've written a really rough timeline, not a timeline, number line for the two means, 7.6 and I should have put 19.1. And I've put in the standard deviations here. Now, to see whether these standard deviations overlap, we clearly have to take 6.5 from 19.1 and add 4.5 to 7.6. And here are the numbers that you get. There is no overlap. 12.1, there is clear blue water between 12.1 and 12.6. Not much, but there is. So doing that maths tells you that those standard deviations don't overlap, and therefore the difference between the means is significant, which is what I wrote. As the difference between the before and after is significant as the standard deviations don't overlap. Actually got that wording right at least. Figure 5 shows that the fetal hemoglobin has, here we go, I'm going to write the same as I did in the previous question, which feels a bit dangerous to me. Generally, if you're writing the same thing in one place or other, you won't get the marks, but I didn't have anything better to write, so I went for it. Higher affinity than adult hemoglobin for oxygen. So a higher concentration of fetal hemoglobin means that more oxygen would be transported. Here's the mark scheme. Now, I gave myself mark point 1, but having read this mark scheme again, I can't give myself mark point 1 because the word used in this mark point is increase. And I just said difference. And that was completely daft on my part because I personally have a rule that if I use the word difference or change, I should be thinking, can I use the word increase or decrease? And I failed to do so, and therefore I lost the mark. Got mark point 2, and I got mark point 3. So I got 2 out of 3 instead of 3 out of 3. And this, I'm going to be honest, this is probably about, you know, this is A01, I would say, terminology, getting terminology right. Question 6. Stomata are found mainly in the underside of leaves of dicotyledonous plant. And a student painted a layer of clear nail varnish on the lower part of the leaf. She peeled off the dry layer, she put it under a microscope, and she was able to see the impressions of the stomata. Hopefully you have all done this within school. She then determined the mean diameter of the stomata. Describe how the student could use an eyepiece graticule to determine the mean diameter of the stomata. Now, if you have no idea of the answer, can I give you a tip to tell you how you can get a mark for any kind of question like this, where it is, describe how, in other words, give me some method, and it has the word mean in it. Because if somebody has got to find a mean value, you can absolutely guarantee that they have to take many measurements in order to calculate the mean. And in this case, the measurements are of the diameter of stomata. So many measurements of stomatal diameter. You've got a mark, and I'll prove that to you in a minute. Here's what I wrote, because I do know the method. The eyepiece graticule is calibrated against the stage micrometer for the objective lens being used. Being a bit of a clever clogs here, because of course that eyepiece graticule, that scale in the eyepiece lens, has to be calibrated for each of the 3 objective lenses in the light microscope. Didn't know whether that detail was going to be needed, but I thought for a few extra words, I can chuck it in. The diameter of many stomata are measured using the eyepiece graticule. And I realized, oh, actually, you know, should I have said that we replaced the stage micrometer with the nail varnish microscope slide? So we did. And the mean calculated, and again, for safety, adding together the diameters and dividing by the number of diameters. Wasn't quite sure where the marks were going to be. Here is the mark scheme. Oh, sorry, I just realized I've got some questions. I will come to them. Here's the mark scheme, and here are the examiner's comments, and this is where I got the marks. So, mark point 1, diameter of many stomata are measured using the eyepiece graticule. The eyepiece graticule is calibrated, mark point 2, and that you take a number of measurements to calculate the mean. Okay, so let's do some questions. Okay, going back to 5.2. Sorry, I just had a question about question 5.2. Let me do that. Right. Okay, so this is, I understand why. So the question is, use figure 5 to explain why this change, in other words, the change from practically totally fetal hemoglobin to adult hemoglobin at 6 months, is an advantage for the baby. Well, the advantage of the change, what they're referring to here is that when the baby, before the baby's born, no, right, sorry, now I've reread it. Right. The advantage of the change from fetal hemoglobin to adult hemoglobin, because the adult hemoglobin, the line has shifted to the right, that means that it has got less affinity for the oxygen, and that means that it is going to find it easier to unload that oxygen to the respiring tissues. So this is about knowing what to emphasize when you are recognizing that a dissociation curve is either shifting right or shifting left. So here we're talking about the change from fetal to adult. The advantage of the baby ending up with adult hemoglobin is that there is increased unloading unloading, dissociation of the oxygen, because there is decreased affinity for the oxygen. Whereas if you were describing the advantage of the fetal hemoglobin, you would say there would be increased association because there is increased affinity for the oxygen. And that is always the way to emphasize the advantage of either an oxygen dissociation curve, either shifting left or shifting right. I understand that when it shifts right, it is harder to associate because there is less affinity. But in evolutionary terms, evolutionary terms is always a bit of a compromise. And basically, the fact that it unloads more easily is more advantageous to the fact that it loads less easily. I hope that helped. I hope so. Yes, so that when the baby is out of the womb, Shri, when the baby is out of the womb, there is greater unloading of the oxygen, and therefore it will be more easy for the tissues to respire. The important thing for the baby when it's in the womb is that it gets the flipping oxygen in the first place from the mother. The fetal hemoglobin curve has to have a higher affinity than the mother's adult hemoglobin. Otherwise, the baby wouldn't get any oxygen in the first place. I hope that makes sense. Hasfa, please do CIE. Right, we have, yes. CIE, really tricky. I'm going to be really honest with you. I don't have easy access to documents with huge numbers of CIE exam questions on, which makes it very hard for me to be an expert on the CIE specification. Can I just say that most exam papers will give you at least an idea of the level of the terminology that you need to be using in any A-level exam paper. I really understand that every syllabus has its, its particular things that it likes. I really get that. But I hope that has helped. So on to, thank you, Sri. On to question 6. Oh yeah, we've done this one, haven't we? Actually, we've done this, and I think I talked about where I got the marks. Yes, okay. Scientists investigated the effect of treating the leaves of one species of plant with 3 different substances. These substances reduce, notice that that word has been emboldened, take note of it on the exam paper, reduce the stomatal diameter. Compared the mean diameter of stomata after being treated with the mean stomatal diameter of controlled leaves that had just been treated with distilled water. In other words, those where the stomatal diameter wasn't going to change. Here are the results. The mean stomatal diameter for the control leaves was 7.5, i.e. this was the distilled water treatment. And this is, notice that this is reduction of the mean. LAM 13, whatever that is, reduced the stomatal diameter the most, then ABA, and PS3 hardly reduced it at all. Calculate the ratio of mean stomatal diameter of leaves. Sorry, let's say that again. Calculate the ratio of mean stomatal diameter of leaves treated with PS3 to those treated with ABA. Notice that they're not talking about the ratio of reduction of stomatal diameter. They're talking about the actual stomatal diameter. So we've got to work out the stomatal diameter. So for PS3, because we know it started at 7.5 and the reduction was 0.4, there's your mean stomatal diameter after treatment with PS3. ABA, similar calculation, the mean stomatal diameter is 4.8. Now we've got to work out the ratio. Here's the ratio written crudely. And notice that I've been careful to get the numbers the right way round because it's PS3 to ABA. So of course, PS3 needs to go on this side to ABA. But the answer requires the second number to be 1 because that is always the convention for writing ratios. And you should always do this if you are simply asked to calculate a ratio. You should always make the second number 1. It is the convention. How do you make the second number 1? Well, by dividing both of them by 4.8. Hopefully that makes sense. It's going to make this number 1, and then you'll have a number, and it is 1.5. The reason I wrote 1.5 is because every number here is only to 1 decimal place, 2 significant figures. Here's the answer, 1.48. But notice the examiner's comments accepted 1.5, for which I was grateful. ABA is a substance that some plant species produce when little water is available. Explain why producing ABA may help these species survive in dry conditions. Well, ABA reduces the stomatal diameter. And hopefully we know what else happens through the stomata, not just gas exchange, but water loss. So if the stomatal diameter is being reduced, the water loss by transpiration will also be reduced. So here's my answer. ABA reduces stomatal diameter and therefore less water will be lost by transpiration. Now, that is what I wrote. Here's the mark scheme. And I only got mark 0.1. And when I look back at my answer, I kind of realized I'd only really written 1 juicy mark point by talking about, oh yeah, I realized that water is lost through the stomata. I don't know whether my brain would have gone to mark 0.2, but it is worth noting. I think then I would kind of go, you know, if I'd realized that I'd only got 1 mark, I should have gone back to reread the question to try and pick up some inspiration as to what to write for my second mark. Explain why producing ABA might help this species. Well, we're helping it by it not losing water. I might have then thought, oh, what would the water be used for? Yeah, photosynthesis, okay. Notice you couldn't respiration, because water isn't required for respiration, it's produced by respiration, so you couldn't write that. Okay, 6.4. Many species of plant can be infected by powdery mildew, which is spread by microscopic spores in the air. Suggest how treatment with LAM13, remember that was the middle column that caused the greatest reduction in stomatal diameter, might protect plants against powdery mildew infection. Well, stomata are holes, and we've been told that these spores are microscopic. They'll go whizzing straight through the holes and therefore infect the plant. So this is what I wrote. So less spores will get into the leaves to infect the plant. And here is the mark scheme, and there's your mark point. Question 7. NMO is a disease that leads to damage in nerve cells in the spinal cord. Person with NMO produces, this is going to get worse, anti-AQP4 antibody that attacks only these nerve cells. And I'm already kind of going, this is an autoimmune disease. Yeah, does it make sense? The antibodies in the patient's body is attacking their own body cells. Not great. Explain why this antibody only damages these cells. We are talking about the specificity of antibodies and why they are specific. So, hopefully you will know that an antibody has specific antigen binding sites. It's those Y-shaped, it's the Y-shaped molecule. Here we go, antigen binding sites. And that they are complementary only to antigens found only on the nerve cells in the spinal cord. Notice how many onlys and specifics you need. Yeah, but I wanted to put in that they were complementary. Antigen antibody complexes. So how does it cause damage? Because we haven't got to the damage bit yet. So now I'm kind of bringing in my immunity topic to understand what antibodies do and therefore why they might cause damage to the body's own cells. Antigen antibody complexes are formed which cause, so am I applying the knowledge that I teach at A level for AQA, because they only really learn about what phagocytes do when antibodies bind to antigens, which cause the phagocytes to destroy the nerve cells as if they were pathogens. So that's what I wrote. I understand as a biology teacher that antibodies work in other ways too. And I'm not at all sure that actually with my slightly greater knowledge that this phagocytes are involved here, but that is beside the point. I'm applying the knowledge that AQA A-level students would have. So here is the mark scheme. And notice you could not use the word active sites, because active sites, yes, they're very specific binding sites, but they are only on enzymes. No other protein molecule has an active site, only enzymes. So here are the marks. I got mark 0.2. It's only complementary to found only complementary to antigens. So I specify the only that are only on the nerve cells, mark 0.3. Antigen antibody complexes are formed which cause the damage. So I got the 3 marks there. I'm kicking myself because I didn't include about the specific tertiary structure of the antibody protein, which of course brings about that specific shape to the antigen binding sites. So that to me is that I missed a basic bit of knowledge that I should have put in. To me, it was an AO1 problem. Terminology. Scientists measured the concentration of these antibodies in the blood of people with NMO. Spinal cord is surrounded by vertebrae. For each person, the scientists determined the number of vertebrae surrounding damaged cells, as well as the concentration of the antibody in the blood. And they produced this graph. It's a scatter graph. Scientists suggested that the concentration of the antibody in a person's blood could be used to predict the number of vertebrae surrounding damaged nerve cells. Use figure 7 to suggest reasons why this may not be valid. So what are all the reasons? What is wrong with the methodology of the experiment? Either to do, are they actually testing what they think they're testing? And are there enough repeats to make the data valid or make this suggestion valid? So to me, the first thing I did actually was count up the number of dots, because every dot is a patient. You've got their number of vertebrae affected and you've got their concentration of antibody in their blood. There's 20 dots here. Now, that is nowhere near a big enough sample size. It's very unlikely. 20 NMO patients are very unlikely to be representative of all patients that have NMO. The sample size is too small. The other thing that I thought of, so here I went back to reread the question because I'm kind of going, not really quite sure what else to write. So they measured the concentration of this antibody in the blood of people with NMO. Well, they didn't say that they repeated it. So I'm kind of going, surely this antibody level concentration could change over time. So I put that down. And then I finally remembered that this is a scatter graph, and therefore what also hasn't happened is that the Spearman rank test hasn't been done to see whether this is significant, whether there is a significant correlation between these two variables. That's what I wrote. I am going to say, mentally, I recognise that there was a lot of variability in the concentration of antibody for particular numbers of vertebrae. Because of my big writing, I didn't have the space to put it in, and I was kind of hoping that my reference to the statistical test would be enough to capture that, because this is 3 marks. I thought I'd done all right. I got mark point 1 and I got mark point 3. I didn't get mark point 2, even though it seems to me to be a reasonable suggestion. But anyway, so I got 2 marks out of 3. 7.3, a new treatment from NMO involves using monoclonal antibodies. Structure of the monoclonal antibody, the variable region is the same as the anti-AQP4 antibody, but the constant region is different. Use this information and your knowledge of antigen-antibody complexes to suggest how this monoclonal antibody prevents the other antibody from damaging the nerve cells. Well, if it's got the same variable regions, it's going to bind to the nerve cells. However, if the constant region is different, that's the bit that actually calls, as it were, phagocytes or whatever, it causes the destruction of the nerve cells. So, oh, and I gave the examiner this abbreviation for monoclonal antibodies. So it stops the other antibody binding. The non-variable region of the monoclonal antibody fails to initiate damage to the nerve cells. So that's what I wrote. Here's the mark scheme. Here's the examiner's comments, and I got mark point 1 and mark point 2. Okay, question 8, home leg here. Pato syndrome is a condition caused by a mutation affecting chromosome numbers. All the cells in the body will have the mutation. Here's the diagram showing the chromosomes from one of the cells of a female suffering from Pato syndrome. What is the effect of Pato syndrome on the chromosomes of this female? If you look at this figure, hopefully you'll notice this here. Chromosome 13 has 3 copies instead of 2, which got me the mark. Describe how the change in chromosome number in Pato syndrome was produced. Well, most obvious thing in my mind is that that has come about from one of the gametes that was used in the fertilization of this female had 2 copies of chromosome 13, whereas the other gamete had the normal 1 copy of chromosome 13. So how did this gamete have 2 copies of chromosome 13? Well, the most obvious thing to me is that in meiosis II, when the chromatids of the chromosome, each chromosome is supposed to separate, that didn't happen. So in meiosis II, one gamete ended up with 2 chromosome 13s, as well as 1 copy of all the others, and the other gamete ended up with no copies of chromosome 13, as well as 1 copy of all the others. So this is what I wrote. And that this gamete, the one with 2, was used in fertilization. Here's the mark scheme. They weren't interested in this last comment, even though I thought it was a reasonable comment to say for this question. I got the 2 marks. Explain why all the cells in the body will have this mutation. So I'm immediately thinking, well, every cell in our body comes originally from the fertilized egg. And the only way the fertilized egg reproduces is by mitosis. And of course, mitosis produces genetically identical cells. And therefore, every cell in the body will have the same chromosome number. Here's what I wrote. Here's the mark points. Here's the examiner's comments. Notice that you could either get mark points 1 or 2, or mark points 3 or 4, but gloriously, mark points 2 and 3 are pretty much exactly the same thing. So I got mark points 3 and 4. Most children born with Pateau syndrome die in the first 12 months, often due to circulation issues. And they often suffer from patent ductus arteriosus, PDA. And these diagrams show what PDA is. Healthy versus PDA child. Notice that the aorta does a U-turn, comes out of the left ventricle and does a U-turn to go back down to the rest of the body. But of course, it is separate from the pulmonary artery, which is where the blood is being taken to the lungs from the right side of the heart. In a patient with PDA, there is a hole between the aorta and the pulmonary artery, and therefore, as this diagram shows, oxygenated blood, because of course the left side of the heart is pumping oxygenated blood to the body. Oxygenated blood is going from the aorta into the pulmonary artery, which is deoxygenated blood. So just how the flow of some of the blood between the aorta and the pulmonary artery could cause children to die in the first 12 months. Well, that is because there is less oxygenated blood going through the aorta, and therefore let's keep on going with this argument. Less oxygen, and I included glucose as well, therefore less oxygen and glucose being delivered to tissues, therefore less aerobic respiration, which would be required for the growth and the development of the newborn baby's tissues. And if that didn't happen, that could lead to the child's death, which is what I wrote. Here's the mark scheme, and here is the examiner's comments again. And so I got mark point 1, mark point 2, mark point 3, and I actually earned mark point 4. Of course, the tariff was only 3 marks, so I only got 3. Final question. 2 big questions, 2 high tariff questions. Describe the cohesion tension theory of water transport in the xylem. So, of course, the emphasis is on what is going on in the xylem. That's where I was reading the question and thinking about explaining cohesion and tension. So I talked about how the water molecules stick together as a result of hydrogen bonds, and that being cohesion. I went into the detail of because water molecules are polar, not quite knowing the extent of detail required. The continuous tube of the xylem from root to leaf therefore causes a continuous column of water. This phrase here I knew was going to be in the answer. It's a phrase that I always use when I talk about transpiration or movement of water up the xylem. This column is pulled up the xylem. Tension, another word for tension is pulling. Think of a tug of war match with the rope. As a direct result of transpiration of water from the leaf stomata. Didn't quite know whether we'd need more information on transpiration, so I gave some more information. And finally, I remembered, okay, there's also the whole thing of the water adhesion to the walls of the xylem vessel. Here's the mark scheme. And I notice you can use water potential instead of writing it out, which is quite useful. And here I got mark point 1. Mark point 3, pulled up the xylem. Mark point 4, stick together because of hydrogen bonds. Mark point 5, continuous column of water. And mark point 6, adhesion. So I got the 5 marks. Slightly kicking myself for missing mark point 2 because I didn't talk about how the evaporation of the leaves links to why water is pulled up the xylem column, and I do wonder whether that wouldn't have been a more obvious thing to write about rather than this answer down here. I got the marks, but I'm kind of making a mental note. I should put that bit in. And finally, describe how mRNA is produced in a plant cell. So the first thing I notice when I read this question is why do they ask about in a plant cell? Well, and then it dawns on me, it's a eukaryotic cell. And of course, in eukaryotes, we get the production of pre-mRNA, and then it's spliced to make mRNA. And then I'm kind of going, oh, I can see why this is a 5-mark question now, because they want the whole transcription detail, and then they want about the splicing of mRNA. So here is what I wrote. DNA in the nucleus is separated into 2 strands because of hydrogen bonds being broken. Free nucleotides, RNA ones, I give the initials, that's a bit risky, probably should have written them out at least first time. By complementary base pairing, they bond to the template strand of the DNA. I did spell out in abbreviations how the bonding happens. And I spent a few seconds working out how to do that very succinctly without writing too much. RNA polymerase then joins the RNA nucleotide by phosphodiester bonds until it reaches the stop base triplet at the end of the gene. The pre-mRNA now detaches from the template strand of the DNA and it is spliced. And that means that the introns are removed and the exons are rejoined. We now have mRNA. Here's the mark scheme, examiner's comments, and here are where I got the marks. And I think I pretty much followed the mark scheme through. I think I got all of them. Yeah, I did get all of them, even though obviously I've only got 5 marks because that's the tariff of the question. And I'm hoping that that has been useful. And I have 4 minutes to spare. Do we have to be specific whether it's meiosis 1? No, you don't, Shri. No, for that, notice in the mark scheme, if you go back and look, the mark scheme only said in meiosis. I knew that it was meiosis 2 that separated the chromatids, because of course meiosis 1 separates homologous chromosomes that are paired up either side of the equator. So that was me, because I knew the detail and it took very little extra writing to put it in. This is where I'm thinking as I'm writing my answers. If I can put a little bit of extra detail in, that's why I use brackets, you know, I use an extra letter, you know, whatever, if it needed to be there to get the mark point, I'd put it in for a little extra time, you know, extra, if that makes sense. No, and also for AQA, you are not required to know that it happens in anaphase of meiosis 2. You don't need to know that either. That isn't a requirement of the AQA spec for meiosis. It is for mitosis. So if you're talking about something going on in mitosis, I would absolutely be naming the stages at which it happened. In meiosis, you are not required to name the stages for the various things that happen. You know, the whole independent assortment, crossing over, all those kinds of things. You do need to know whether they're meiosis 1 or meiosis 2, but that's it. I hope that helps. All right, I'm just seeing your answer. How do I know how detailed I should answer the question and not miss points? Right, I'm just going to say to you, that is practicing answering questions. And you will get to know some of the things that are going to be in the mark scheme, which is sometimes why I said I kicked myself for missing a mark point. And other times I kind of went, do you know, I know I've got plenty of marks to be, you know, for me as a teacher to be on track for an A star, to be up, you know, close to 90% of the marks. Do you know what I mean? Or over. And And to be quite frank, that's all I'm worried about. If I drop a few marks and I look at the mark scheme and kind of go, just wasn't the way my mind worked. As long as I've got enough marks to get the grade that I'm aiming for, I'm not going to get too hung up about it. What of course you need to make sure that you're doing is that your brain is thinking along the right lines for enough of the mark points for the grade that you need. And that is about clearly knowing your content, knowing your terminology. Go back to my revision, have a look at my revision video for that. But also then practicing the exam questions so that you get to know the terminology and the phrases that come up time and time and time again. Hopefully that helps. Candy, do you have advice with regards to synaptic assay on paper 3? Yes, I've done a whole video on it. I can't remember whether it's public on my YouTube channel. If it isn't, it is on my website, www.biologycarol.co.uk. I can't do that now. Loads of love to you guys. It's now 6 o'clock. I need to go off and tutor, so another hour of talking. Lovely to see you. Thank you for joining me, and see you next Tuesday. Bye-bye.